So much of the conversation about CRISPR has revolved around the potential for treating disease or editing genes in human embryos, something we're gonna discuss. But researchers say the real revolution right now is in the lab, and you're a bench scientist, you've been working with mosquitoes. Tell us what you do and how CRISPR is involved in that.
Sure, absolutely. So, as you said, I work directly on mosquitoes, which previously have not been really a genetic model, meaning that we can't edit and engineer their genomes directly until the advent of tools like zinc finger nucleases and TALENs, and now CRISPR. And so what CRISPR lets us do is actually mutate genes in the mosquito that we think are responsible for their system of smell and taste. And so what we are interested in specifically is figuring out how a mosquito in its environment will find you, for example, and take a blood meal. And so what you're looking at up here is a female Aedes aegypti mosquito, and so she is the vector for a number of arboviruses, including Zika virus, which is a big problem these days, and some that have been around a little bit longer, like dengue fever, yellow fever, and chikungunya. And so a female mosquito is exquisitely tuned to find and bite human beings. And females do this because they need to develop eggs. So that blood meal that you see her taking right there is going to about double her body weight. And so she is going to drink about 2 milligrams of blood, and then she's going to turn that into a batch of 100 mosquitoes. And so this is why evolution has really acted on their sense of smell and taste. And so what we do with CRISPR, and this slide is just kind of schematizing how we do it. So we have taken these components that George and others figured out in bacteria are responsible for cutting DNA at specific places, and we re-engineer them in the lab and then re-inject them back into mosquito embryos. And we do this not to make that embryo mutant, but we do it so that we can mutate a sperm or an egg in that embryo, which is going to give rise to a stable mosquito mutant. In essence. So it's a mosquito that either is missing a certain gene, or we've engineered a certain mutation in a gene, or what we're doing now, which we're really excited about, is actually introducing new sequence. And so what we can do now is we can get genes that we want to express in cells that we want to have those genes expressed in, and we can get control over neural circuits. We can image the brain of the mosquito as it's hunting for a host in the environment. And so this is showing you just what happens when we put in a sequence of DNA in a particular locus. So you have a blue and a red mutant strain of mosquitoes. Each one of them is missing a gene that we think is involved in their ability to find an appropriate place to lay eggs. And what we've done is we've actually replaced that gene with a ubiquitous fluorescent reporter, so either a blue or a red protein that we can see under a microscope. And so we do this so that we can follow them in the lab, and then we assess their behavior with really sensitive behavioral assays to identify which of these genes are involved in the critical behaviors for the mosquito's life cycle and disease transmission. And so this has really been the result of the last 2 or 3 years. We've come much, much further than we had in the previous 5 or 10, specifically because of the advent of CRISPR. And so my experience with people who work on other non-model organisms is that if you can figure out how to get these reagents into an embryo, CRISPR will work. It will make its cut. And that didn't have to be the case, and so that's one of the most amazing things, to me at least, is that something that's so evolutionarily ancient will work in human cells or mouse cells or mosquito cells. And so what we do, and this is my hand, so we're not going to release these anytime soon into the environment, but you can imagine that by generating certain strains of mutant mosquitoes that may be poor at targeting human beings or have other altered behaviors, that we can understand the processes that they use to find and bite people and by proxy transmit disease.